Hi, everyone. Thanks for joining us. We'll be starting in a few minutes. Okay, great. So thank you everyone who's tuned into our YouTube live stream for this event. We are ready to begin. My name is Kate Richards. I'm Director of Communications at the Leslie Dan Faculty of Pharmacy. On the screen, you'll also see we have our expert speaker, Dr. Keith Pardee, as well as two postdoctoral fellows from the Pardee Lab, Dr. Margot Kolikoff and Dr. Masoud Nourouzi, who will be participating in the Q&A session following Dr. Pardee's talk. When I joined the Leslie Dan Faculty of Pharmacy about 2.5 years ago, I came with experience in healthcare and research communications, largely in the hospital sector and primarily in mental health. Before joining the faculty, I had never encountered the term synthetic biology. Since then, I have had the pleasure and the honor to work with Keith and many members of his lab to help communicate about their incredible work developing rapid diagnostics for infectious diseases. Fast forward to today, and we all find ourselves in the midst of a global pandemic. In many ways, the pandemic has propelled the pharmaceutical sciences and scientific work, like that being led by the Pardee Lab, to the forefront of public consciousness. While I imagine many people who have registered for this talk are bringing significant scientific expertise in pharmacy, healthcare, biomedical sciences, we also hope there are folks who are perhaps more on my end of the spectrum, who are not scientific experts, but understand and appreciate the urgent importance of this work. I encourage you to also ask questions during the Q&A through the YouTube chat function. Before I hand over to Keith, I would like to provide a very brief overview of Dr. Pardee and his work. Keith is an assistant professor and principal investigator in the Department of Pharmaceutical Sciences at the University of Toronto's Leslie Dan Faculty of Pharmacy. He is also a member of PRIME, a new cross-divisional initiative at U of T focused on next-generation precision medicine. Keith holds a Canada Research Chair in Synthetic Biology and Human Health. He is the recipient of numerous awards and distinctions, including the U.S. Office of Naval Research Visiting Scientist Program 2019, Emerging Leaders in Biosecurity Fellow, Johns Hopkins University 2018, New Investigator Research Awards, Association of Faculties of Pharmacy of Canada 2017. Keith leads a highly productive lab of established and emerging scientists. The Pardee Lab seeks to impact human health by developing portable, affordable tools using the principles of its synthetic biology. In March, the Pardee Lab was awarded $1 million in research funding support by the Canadian federal government to further pursue his lab-in-a-box rapid diagnostic technology in the context of COVID-19. Keith, thank you for hosting this talk. Please begin when you are ready.
Great. Well, thank you very much, Kate, and thank you all for joining us today to hear our work. We're really excited to share our work on COVID-19, as Kate mentioned. Joining me today is Margot and Masoud, and who have been working tirelessly in the lab since this all began. And so they'll be joining me at the end of the talk to answer questions. So thank you very much, guys. They deserve a tremendous amount of credit for all the hard work they've been doing. And so with that, I'll dive into the talk, and then we'll do questions at the end. So let's share my screen. All right, so there we go. Great. Okay, so thank you very much. as Kate mentioned, we work in the field of synthetic biology, specifically cell-free synthetic biology. And what is cell-free synthetic biology? What we basically do is we take the gene circuit out of a cell, which is where gene circuits are usually run, and we run them in an in vitro environment. And that allows us to do a couple important things. For one, by running in this in vitro cell-free environment, it's biosafe, so it can't replicate, but it can still run the gene circuit. And this lets us take these tools out of the lab in a biosafe way. And it also lets us freeze dry the tools so that we can distribute with at room temperature without refrigeration. And so we use this platform, this freeze-dried cell-free platform, to make tools that make healthcare more accessible. And so we do that in two main areas. One is in portable sensing, which is the topic of what we're going to talk about today. And the other is portable manufacturing of protein-based drugs, such as antibodies and vaccines, which is also, you can imagine, also very relevant right now. But today we're going to focus, and most of our lab right now is at this point focused on diagnostics. So I'm going to start off with some background on diagnostics and our Paperbased platform. I'm sure many of you, although I can't see you, are scientists, but there may be some that are more generalist, and so I wanted to provide some background there. And then I'm going to talk about how our platform how we've used it for Zika virus and the subsequent patient trial that we did for the Zika virus diagnostic, and our current work on SARS-CoV-2 and the next steps for the project. So we've all become sort of amateur experts in public health over the last few months. And we all know that sort of to get out the other side of this, we need a vaccine, right? And so, but unfortunately on timescales, that's going to take months to years. And so we need measures to carry us forward and safely through that to prevent infection and spread of the virus in the meantime. And so on sort of the immediate timescales, social distancing and personal protective equipment like masks when needed are immediately available pretty much, right? We all became quite good at social distancing in a matter of days once we sort of flipped the switch. A little longer timescales are diagnostics, and diagnostic access has been something that's been in the news a lot. And so as you've seen, right, this capacity for diagnostic throughput has been spooling up over the last few weeks, and we're now significantly in Ontario and globally doing many more tests than we had at the beginning. And as we sort of proceed down this path, it's going to be more and more capacity and more diversity in those tests. And so social distancing and diagnostics are going to be key public health tools as we sort of work towards the end of this crisis, which will be a vaccine, or, you know, as we saw with SARS-1 or Zika, it can also just sort of wind down even without a vaccine. And so our diagnostics in general are going to be really important in restarting our economy and just returning our lives to normal. So I wanted to briefly touch on the different types of diagnostics that are available. So there are, there we go, two types, touching my screen. So there are two major types. So three major types. The first type is an antibody-based test. And this is a test where it has an antibody specific to the virus, and the antibody recognizes the virus. And that's what that pregnancy test is based on this. And often these are lateral flow assays where you get a band if there's a positive. There's also serological tests. These are really important, especially probably in the future, where we're going to be looking for who's already immune as we sparingly dole out our vaccine with most critical people involved in infrastructure and health, getting the vaccines first and making sure that we're using the vaccine as efficiently as we can and ensuring that people, as they return to work, are in fact have antibodies for the virus and are therefore immune. And the third category of diagnostic is the molecular diagnostic. And this is a diagnostic, this is a tool that looks for the genome of the virus. Essentially, the genome of the virus is the barcode that we look for and identify. And the most, the only molecular diagnostic being used in clinical labs right now is something you've all probably heard of, RT-qPCR. And so that stands for real-time quantitative polymerase chain reaction. And thought I'd share a little bit about that with you guys, just in case you're not familiar. And so how it works is you somehow open the virus up using chemicals or heat, and that exposes the genome of the virus. And then through a cycle of heating and cooling steps, enzymes replicate and make millions and millions of copies of a very specific region of the target region in the genome. And you get, as that target region accumulates and amplifies, you get an optical signal that can be measured by the PCR machine, and that would indicate a positive test. So our lab, let's sort of set the context of what our lab does. So our lab is largely dedicated to using the Cellfree platform for decentralizing tools for healthcare. And in many cases, we're doing that for low and middle income countries where healthcare access is limited. And so we do this by developing gene circuit-based diagnostics. It's a relatively new approach that's been pioneered by our lab and others over the last 5 years. And one of the main advantages is it's very low cost to develop, and also manufacturing costs are quite low. And both of these translate to better accessibility. And so we're applying our gene circuit-based diagnostics for food security, emergency preparedness, like we're going to talk about today, general health care, right, and global health, and bioresilience, or resilience to biothreats. So in working towards these diagnostics for often in low and middle income countries, the needs of those countries, we've designed our diagnostics with some unique features. Some of those, that includes being independent from the PCR supply chain and instrumentation, because these are generally more costly. And instrumentation can be hard to find, and it's bulky and not portable. We've also developed our system so that the molecular products that are used to make the diagnostic can be manufactured locally. And so this gives us some resilience to supply chain disruption. And then we've also developed methods that allow for silica-free RNA extraction for diagnostics. And so for many of you, these features may seem very familiar in discussions with diagnostics around COVID-19, because all of these factors are now globally relevant as the supply chain for PCR has tightened and been strained, as have the other reagents like RNA extraction kits. And so we felt, well, we have a lot to potentially offer in this field. So I'm now going to switch over to cell-free synthetic biology and just provide a bit of background on the general field and how we approach and build our tools. So as I mentioned, we take the enzymes out of the cell that are these are enzymes of transcription and translation, and these are the enzymes needed to run a gene circuit. And so we use the enzymes from E. coli, but often the other labs can also use other bacteria or eukaryotic systems like tobacco or yeast. The other major component is DNA that encodes for our sensor, our gene circuit-based sensor. And it basically serves as a unit, as a DNA encoded tool that can sense and then produce an optical output in response to the detection of that analyte. We then freeze dry these components so that they can be distributed at room temperature without a cold chain. And in one embodiment, we'll embed those components into paper. And this lets you have this physical ticket that you can hold and run your test with. And so what you're looking at here are two paper discs that are 2 millimeters across, and they sit in a 384 well plate. and they have the enzymes of transcription and translation. The disc on the right has the DNA encoding for green fluorescent protein from jellyfish. And so after rehydration and incubation at 37 degrees for 20 to 30 minutes, you start to see expression of this green fluorescent protein. The other embodiment is paper-free. And here we take this cell-free reagent and we create pellets. And upon rehydration, we can express proteins. And so in this case, we're expressing GFP, the fluorescent protein. But in practice, what we do is we're producing vaccines and antibodies in this format. And the focus of today's talk is on the sensor side of the equation. So that paper-based sensor-free, cell-free system is essentially an operating system. And it needs software to run to create those sensors. And so for that, we turn to RNA-based components. And specifically in the project we're going to talk about today, we use a Toehold switch. And so Toehold switch was invented by Alex Green while he was doing his postdoc with Peng Yin and Jim Collins in Boston, where we met. We were both at the Wyss Institute. And a Toehold switch is an RNA hairpin that sits at the 5' end of an mRNA, and it sequesters the ribosomal binding site. And in doing that, ribosomes cannot bind, and therefore you don't get translation from the RNA into protein. And this region marked AB is effectively the sensor surface. And so if you want to detect your trigger RNA, you design the AB region to be the complement to that region. And so A star binds to A, B star binds to B, and you get linearization of the hairpin, ribosome combined, and you get translation of your protein into your reporter. And so what this effectively is a rational approach to designing diagnostics. And so we can design diagnostics against virtually any system or any pathogen with this approach. But we needed some more features to make this practical outside of the lab. And so for distribution without refrigeration, we turned to freeze drying, as I mentioned before. In low resource settings where you want to know what's going on at the molecular level with your eye alone without any instrumentation, we needed to create color-based outputs. And so we've done that. And then for more sophisticated applications, we've developed low-cost electronics that allow for quantification and potentially automation of processes. And so our first paper back in 2014, we sort of laid the groundwork. And so we developed the color-based outputs so that when the Toehold switch sees its target, it creates an enzyme that creates a nice dark purple color. We created a way to multiplex sensors, so sort of spotted arrays, and then hardware for low-cost hardware for measurement of the reactions. And so we applied this as in demos for the detection of two things, antibiotic resistance genes, and also strain-specific detection of the Ebola virus. And so at the time, this was really exciting. This was the first time you could take these cell-free, these gene circuits out of the lab in a biosafe and paper sort of embedded format. They weren't sensitive enough to be clinically relevant or useful. And so we spent the next year tackling that challenge. And that's when I finished my postdoc and came to the University of Toronto. And as did Alex. He went on to his respective university at Arizona State. And then upon about a month into my being studying at the University of Toronto, the Zika outbreak happened in Latin America. And so Jim called Alex and I up, and we agreed that we'd taken the platform far enough along to contribute to this outbreak. And so the first step was for Alex to run the genome of the pathogen through his algorithm. And this is one of the first things that now happens when there's an outbreak. Countries generally post a sequence of the pathogen that's responsible for an outbreak online. It's publicly available, and this becomes our raw material for building sensors. And so Alex's algorithm looks for regions of the genome that are available and not folded with secondary structure, so that the Toehold or other sensors can bind to that region. And that complement is then placed into the Toehold switch. And that's how we build our sensors. And so this is a relatively low cost approach to developing sensors. And then the manufacturing costs are also quite low. And so this is work from many people, many institutes, and it was led out of Jim Collins' lab. And I just want to recognize all of those folks before I share the work about this. Yeah. So to solve the sensitivity issue, what we did was we placed an isothermal amplification loop in front of the Toehold switch. And so this is a primer-directed method where primers specific to the region you want to target bind to the RNA, and then through reverse transcription, you get an RNA-DNA duplex. Then RNase H is there. It degrades the RNA bound to the DNA. So now you just have a single-stranded piece of DNA. A second primer comes in with a T7 promoter on it. And through double-stranded DNA synthesis, you get a piece of double-stranded DNA with a T7 promoter, which essentially is a RNA generator. And so this goes around and around, and you get on the order of tens of millions of copies of amplification. And this is what's used to amplify or to go and hydrate your paper disc. And so this gives us sensitivity down into the low femtomolar range, which is clinically relevant for Zika. And importantly, we were also able to get this down into the low attomolar range and even the mid-zeptomolar range. So sensitivity with this approach was quite doable. And so we then tested for specificity. So we challenged our Zika sensor to discriminate from RNA from cultured Zika virus, from cultured dengue virus, and we were able to do that. And then with samples provided by Dave O'Connor at UW-Madison, who was studying the effects of the Zika virus on macaques, and who gladly shared some infected serum with us. With a 1 hour, with a isothermal amplification pre-step, we were then able to detect the virus and discriminate from controls using in-house hardware. And you can see this chip where you have control and reactive spots. And so in the end, we were able to build a sensor that could detect all the global strains of the virus with this and seeing this nice deep purple color, but distinguish from other closely related viruses or arboviruses like dengue. And then using a CRISPR strategy developed by Guillaume Lambert, who's now at Cornell, we were also able to do single base pair resolution distinction there, which was important for the Zika virus because of microcephaly associated with the American strain of the virus. And so in the end, we had this, we were able to have clinically relevant sensitivity, but importantly also developed a programmable platform. And so that it could be pointed towards any future needs and outbreaks. And we're going to get to that shortly with the COVID related work. We were then very fortunate to have the opportunity and the funding from CIHR and the IDRC in Canada to take and establish a field trial and patient trial in Latin America. And so as a group of 8 labs from 5 countries and our Latin American partners in Brazil, Ecuador, and Colombia, we set out to do a patient trial of this diagnostic, which was one of the first of a synthetic biology technology being done out in the field. And so I'm going to share data that we haven't really presented yet with you on this, and because the trial's recently wrapped up. And I'm also going to talk about some hardware that we've had to develop. And so we've now become not just a molecular lab, but also a hardware lab. And so for the development or for the practical distribution of these tools, we need to develop companion hardware for applications outside of the lab. And so in 2018, we were down in Ecuador where we brought this hardware down along with molecular tools, and we trained our partners from Colombia, Brazil, and Ecuador. Everyone came to Quito for workshops and training. We got to all meet each other face to face. And then last year in April, all of us again descended on this time at Recife in Brazil, which was the epicenter of the Zika outbreak. And we started the patient trial. And so that work was done while we were there and continued on well after we left by the group in Brazil at Fiocruz. And so this is the hardware. So this is basically, you could think of it as a portable plate reader. It's about the size of a toaster. It's got incubation, so it runs at whatever temperature you need. And we call it Plum. And so we've developed the user interface. So if you're used to using a plate reader, it's very similar to that. So you've got a map where you can set all your reaction identifications. And then it also has data analysis on board. And so, in fact, this data analysis process can automatically distinguish between positive and negative samples, and then potentially that's quite useful down the road as we aim to sort of distribute this capacity out. And so it can distinguish from this with this threshold line here. And so through Wi-Fi connections, we can also remote in, or we can see data streaming as experiments are being done in Brazil, here in Toronto. It has the option of being battery operated, so you could literally throw it in your truck and go do some testing somewhere. And so in principle, or basically in sum, this is a really important tool for provided distributed high capacity testing. And this work was done by Livia Gu and Sarai Chichek in my lab, who are dynamos and who are now commercializing this technology. And I'm going to share a link to that at the end of my talk. And they're doing really great work. So the project, we then use the Plum reader to do our sort of validate the diagnostics. And so the first step to that was determining specificity. And so the data on the left is the data that was read on the Plum instrument and done with Toehold switches. And the data on the right is the RT-qPCR test from the CDC. So it's our gold standard that we're comparing to. And so you can see this purple color was only found in the well that contained the Zika virus. And viruses, so Mayura, chikungunya, yellow fever, 4 isotypes or serotypes of dengue, and the water control all were negative. And we see the same thing on the RT-qPCR side, where everything is negative except for Zika. And so our assay or diagnostic platform here had equivalent specificity to the RT-qPCR test. The next step was to determine specificity. And so here we took a dilution of cultured Zika virus from 10 to the 4 to 10 to the negative 3 plaque-forming units per mil. And we ran the assay again. Again, our data for the Toehold switch is on the left, and the RT-qPCR data for the CDC Zika test is on the right. And so you can see that we could detect down to 10 to the 1, which matches what we see with RT-qPCR. So similarly, the sensitivity of our assay was equal to the CDC's RT-qPCR Zika test. And then we went on to run our patient trial. And so this was done with 300 patients with a Zika prevalence in those samples of about 28%. And our platform provided 98.5% accuracy. So we were thrilled with this outcome. This was a very challenging project, but we were really thrilled with the outcome. And it really set the stage for what we're doing now with COVID-19. And so when the outbreak first happened over the holidays, we didn't really pay much attention, but it did get our attention in early January when we were starting to see some of the severity of the outcome. And so we started collaborating with Alex Green again and started working on this. And so by late January, we had sort of committed to doing this. And we'd written a grant. So the CHR, IDRC, and the tri-councils in Canada were very responsive to the outbreak and put a call out for grants very early on compared to most other countries. And so we put a project together. At this point, the outbreak was confined largely to China. And so we put a grant together for developing tools for low and middle income countries, where the World Health Organization had identified these countries as potentially vulnerable to the outbreak. And so our proposal included the development of sensors for SARS-CoV-2, a deployable lab-in-a-box kit that basically would allow you to instantly sort of deploy surge capacity anywhere you needed it. And then a third component where we developed the infrastructure for on-site fabrication of kits so that we could sustain that diagnostic capacity. And we did this with our collaborators in Brazil and Vietnam. Now, fast forward a month and a half, as we're all too painfully aware, the virus rapidly spread, and it's gotten far worse since this time point. And so now we've adapted our project to also include validation and patient trials here in Toronto. And so our project has three main components, and we're focused right now on this step here, which is the development of SARS-CoV diagnostic sensors and the validation of those. The next is this kit for surge capacity. At the time when I wrote this grant, I thought 14,000 tests was enormous amount of tests, but now it seems in retrospect like a drop in the bucket. And so we designed this test to provide 1,000 tests per day for 2 weeks, this kit. And then a third box or component, which is for on-site fabrication of the tests so that the test diagnostic capacity could be sustained. And so these are low cost and deployable, and importantly can be produced locally. And so that it builds resilience to supply chain disruption and is independent of the strained PCR supply chain. And so the team is growing. This is the original team that was on that first grant, but it's certainly grown over time. We're now happily, we're getting a lot of collaborators joining us to help out. And so the project is with Alex Green's group at Arizona State, the Canadian Food Inspection Agency, and Public Health Agency of Canada in Winnipeg at the National Lab. We're working with Lindemar, who's at Fiocruz in Brazil, and Mai, who's at the NIHE, the National Health System in Hanoi in Vietnam. On campus, we're working with Jinyu, who is a mechanical engineer, who's helping us port our diagnostic capacity into a point of care platform. And then in my lab, Margot and Masood are sort of doing everything, right? So they're the ones who've been in the lab this whole time pushing this platform forward. And so all the data that you're going to see is come from their hands over the last month and a half. So when Alex designed the Toehold switches and sensors for SARS-CoV-2, we targeted 6 regions in the genome. The genome 30,000 base pairs, which is quite large for a virus. And we have sensors in the open reading frame 1, the polymerase gene, the envelope protein gene, and the nucleocapsid gene N. And so Alex has designed one set of 24 sensors to each of these regions. So 24 sensors to the polymerase, 2 sets of 24 sensors to the envelope gene for envelope protein, and 3 sets of 24 sensors to the nucleocapsid gene. And so the complement to those regions is placed in this AB region, and in the presence of the correct sequence, we get expression of our reporter, which is LAC-Z, creating that nice purple color. So just to provide a little bit of overview here, we've got the computational design, then the Toehold goes in the paper along with Cellfree, and then upstream we have the isothermal reaction. And so if the COVID-19 or the SARS-CoV-2 genome is present, you'll get a change in color, and this occurs inside a plate reader. And so Masood started assembling these genes quite a while ago, actually, and had very rapid success. We were able to, so what you're looking at here is optical screening that's done on a plate reader. The DNA was from PCR assembly, so it was directly from PCR cleanup step and directly into cell-free, which allows basically a cell-free cloning step and allows us to go much faster than normal. The DNA for coding for the RNA was also supplied as template. And so the output, like I said, was LAC-Z. This creates the purple color, which we monitor at 570 nanometers. And I guess one thing I should mention, I forgot to mention at the beginning, is the data that I'm sharing with you guys is very much in progress, right? This is sort of, you're watching real-time science. And so this is all still very much in progress and not complete at all. But wanting, in the spirit of wanting to share as much as we can, we're presenting it today. And so the brick colored line is the reactions that contain the synthetic RNA that code for various respective regions of the SARS-CoV-2 genome. And the green line is the control. And you can see that some of these sensors worked remarkably well and quite quickly. So the next challenge was to develop sort of the sensitivity, to get it into the range that's clinically relevant. And so again, we turn to NASBA. And so basically in the workflow here, we have a NASBA step that takes that RNA input, amplifies it. Then in that multi-well plate, we have a combination of reporter, paper, and reagents that make the color change. And this is all running in the plate reader and monitored over time in the plate reader. And so Margot's been working on this, and to date, she has got the sensitivity down into the low femtomolar range, which is what we had for the Zika virus originally, and is certainly in the clinically relevant range. And Margot and Masoud are continuing to push this sensitivity down further into the attomolar range this week and onwards. An important sort of consideration is that, as with any pathogen, they mutate a lot, or quite rapidly at a rate much more frequent than other organisms. And so there's been over 500 mutations documented already for SARS-CoV-2, and you can imagine that some of these will be clinically relevant. And so being able to track viral strains with single base pair resolution could be quite important. And so with work that just came out of Alex Green's group again, at the beginning of March, he developed a new type of riboregulator called a SNIPER, that stands for single nucleotide specific programmable riboregulators. And they're very similar to Toeholds, but they have this energy balancing region here that's very sensitive to mutation. And so if you have the correct sequence, you get unfolding and you get translation of your reporter, just as you would with a Toehold. But in the presence of a mutant, a single base pair mutation, you do not. And so so we thought this would be a very important thing to do. And so Masoud's been also madly working on building these snipers, and they'll be important, also for distinguishing against other potential coronaviruses that are in circulation. And so this is data from his first round of testing of the snipers. And you can see that using RNA to specific region of the COVID-2 genome, and those homologous regions in CoV-1, which is the SARS-1 strain from 2003, and MERS, which is another coronavirus, we get very specific detection. And of course, CoV-1 is not in circulation, but it potentially could be important to be able to distinguish between the strains. And so in data that Masoud's just completed over the last couple of days, he's gone and done a much more sort of intensive look at this and compared in triplicate Toehold performance to sniper performance. And so here you can see that, let's look at this one, right? So here the Toehold switch was actually very specific against CoV-1 and MERS on its own, but not the case for gene E, where there's less diversity. They're very closely, these genes are very closely related in CoV-1 and CoV-2. And so you can see that while we don't pick up MERS, this sensor does in fact pick up CoV-1. And so with the sniper though, which is in snipers, which are in orange, there's a remarkable lowering of the background, right? You can see very clean background and low background. And so we're really excited about the implementation of these snipers into the workflow. And so now we find ourselves in mid to late April. Time is flying and going slow at the same time. And we're at a point where we're continuing to optimize sensitivity for our sensors. RNA from the National Lab in Winnipeg will be arriving tomorrow, so that we can do our specificity testing. This will include full genomes of SARS-1, MERS, and 2 influenza genomes. We've been very fortunate to partner with a team at St. Michael's Hospital for patient samples. And so we'll be starting our patient trial in the coming week or two with them. And if anyone who's on this call knows someone who has patient samples, we're also keenly interested in talking to them as well, because we see many rounds of having to do patient trials going forward as we move this technology out into use. We've already talked to Health Canada, and we are planning to immediately submit an application for approval with our patient trial data once we're done. And recognizing that scientists have different skill sets than people that take things to market, we've formed a not-for-profit that's dedicated to scaling and distributing these tests outside of the university. And so this will be basically this. The lab's going to serve as the sort of research engine, and we'll hand the technology off for scale up and distribution to this entity that's called Incarta. And I'm going to share a little bit more about that shortly. In June and afterwards, we'll start the testing of these kits in Brazil and Vietnam with our partners there. And we'll start working on developing the local sort of kit fabrication or diagnostic fabrication protocols. And so, I think just like as a window into the world of research right now, you can imagine at home, it's challenging simply just to buy groceries. And so those challenges are amplified in the lab many-fold. So, ordering is challenging because many of the vendors are either under strain or understaffed at the moment. The buildings are locked, and so coordinating shipments is hard. But it's working, right? It's all working. And so, and then of course with social distancing, the lab is certainly not operating at a capacity that's sort of a level that's normal. And so just when you want to go faster than ever, you face these challenges, but Masood and Margot are making incredible strides, and I'm really excited about where we're going. So we'll be there soon. And so my acknowledgements, Masood and Margot have done an incredible amount of work here, and we're going to get a chance to talk to them shortly. Alex Green in Arizona and his designs with his team have been really important to the project, and he's a key collaborator. The team in Winnipeg at the National Lab, Lindemar and Jefferson and Fiocruz in Brazil, Mai in Vietnam, and Jinyu here on campus in Toronto, who will be helping us, who's already started working towards converting these tests into a point of care tool. The work I presented for the Zika diagnostics was out of Jim's lab, and these are all of the people that contributed to that project. And of course, I'd really like to acknowledge the support from IDRC and CIHR for this work, and most recently, a grant from the University of Toronto's COVID initiative as well. And lastly, I also want to give a shout out to LSK, who is Livia and Sarai and myself, who are, but really Livia and Sarai, who are championing Plum and taking it out to market at exactly the right time, where it can be very useful and empowering for the response to COVID. So our non-for-profit is called Incarta, which is in paper in Latin, and is comprised of 5 founding members: Guillaume, Margot, Masood, Alex, and myself. We are recruiting team members who have skill sets in sort of engineering and process and operations and sales and business, all the skill sets that scientists maybe don't have as much as, and we're also actively raising funds. And so if you're interested at all in this effort, please contact us through the email that's here. And with that, thank you very much. And I will stop sharing my screen, and we'll have Masood and Margot join us. So thank you very much.
Thank you, Keith, for that amazing talk. We do have some questions coming in on the YouTube chat. So I'm just going to let people know that we're open for Q&A. We did have a few questions in advance, actually, from a few kind of, keen people. So I'm going to start with one of those questions. And if a question comes in that I read to you that you'd like to hand off to either Margot or Masood, please go ahead and do that. So an early question that we got prior to the webinar was, when did you start thinking about how your lab could do COVID-related research? What is it like to pivot your research program so quickly?
Sure. So yeah, I mean, I would say mid-January, it became something that we became very interested in and started talking to our collaborators at the National Lab in Winnipeg. And very shortly after, Alex designed the sensors and Masood started building. So, but I don't know, Masood, maybe you could share your thoughts on what it was like to basically have to drop everything and transition, or your experiences, both of you guys, your experiences in sort of, you both had to really change gears from what you were doing. You're certainly not doing what we're doing now in December. Yeah.
Yeah, basically since mid-January, when we started to basically get in touch with the collaborators and asking Alex Green to design the sensors. And as soon as we received the sensors, it was basically most of the time in the lab was dedicated to basically fast tracking and building the sensors. And also since the university went into shutdown like a month or 5 weeks ago, it has been 100% dedicated to basically trying to develop, screen, and optimize the detection protocols for the sensors. But luckily, we are backed by years of experience from Keith, Margot, and the collaborators. So it is it is not something that we don't know what's going to happen. We are confident that we will have a fully functioning set of sensors.
Yeah, I guess to add to that, what we've done and deployed for Zika is almost identical to what we're doing here. It's just the primers for the NASBA, which these guys are tirelessly working on now, and the Toehold switch. Everything else can stay the same. So it's really a programmable platform.
That's great. And one thing that struck me in your presentation was when you said you were putting together the proposal for the CIHR funding and thought that 14,000 kits would be, a huge amount. And now, only 4 or 5 weeks later, we're looking back and you're right, we're seeing that that's a drop in the bucket. So it's incredible to see how much has changed in that short time. I do have a couple questions coming in through the chat. So I'll read one here. This one's from Michael. It says, awesome presentation, Keith. Really cool technology and lots of hard work from Margot and Masood. For the sniper switches, do you add in many different switches for the same gene? Do you run the risk of missing detection in a mutated SARS-CoV-2?
This is a great question. So I'll answer one part and then let Masood answer the rest. But absolutely, we'd be definitely at risk of missing something if we just used a sniper because of the mutation rate. And so we would pair a Toehold switch, which is much more tolerant to mutation. So a Toehold switch can tolerate over 10% mutation and still be triggered. And so we would pair those up. But Masood, maybe you could sort of talk about how you screen the
yeah. For the initial screen, just to identify, basically quantify the performance of each individual switch, we do individually. But in the end, once we have a set of validated switches which are specific for the basically COVID genome, we have the option, and also it would be a good choice to add more than one switch to the same reaction. But for the initial screening, it is only one switch. But for the practical application, it could be more than one.
And I guess, Margot, there's precedent for being able to do multiplexed NASBA, right? I think.
Yeah. Yes.
Yeah.
With multiplex NASBA primers, we could target 2 regions of the genome that would match 2 different snipers. And as much as the virus is mutating, I think we would drastically reduce the chance to not detect the SARS-CoV-2 with 2 snipers that would match different genome region. So I think we would be covered. Yeah.
Cool.
Great. So I have another question here from the chat from Eliza. And she says, I'm wondering if you are anticipating needing greater sensitivity than for Zika, given the wide range of symptom severity with COVID-19.
So based on the literature to date, the viral titer in patient samples ranges from 10 to the 4 to 10 to the 7 viral copies per mil. And that corresponds to low attomolar to low femtomolar. And so if we can get, basically, if we can match what we did with Zika, we should be able to get most of the clinical sample sort of concentration ranges. Of course, in early stages of infection, like any technology, there's challenges, right? PCR also probably likely miss the very early stages where viral titers are low. But I think Margot's experience with NASBA, she's been able to get it extremely low sensitivities. And
so I think sensitivity is mainly due to the primer combination you're using. And with Zika, I was able to go like zeptomolar, as Keith mentioned. Also, I think people being tested right now are those that are going into ICU that have symptoms. And in the most common test, which is the nasopharyngeal, swab, it's where you can find the highest titer right now. So with those combination of good primers that we're currently working on with Masood, and also those patient samples, those swabs, I think we should be able to detect coronavirus, like good sensitivity.
Yeah.
And it was incredible to hear. It was news to me that you said that that pathogens mutate very rapidly, and that there has been 500 mutations documented so far, which, as a member of the general public like myself, I actually didn't know that. So I can imagine, what a challenge that must be. We do have another question from Andy that says, one question is, how designable, in quotation marks, are the toe switch sequences? Are there limits, or can it be virtually limitless, similar to CRISPR?
Why don't I let Masood or Margot answer that? I mean, yeah.
I could say that it is limitless, actually.
Yeah.
That's a good thing about Toehold, is that basically you can put virtually any sequence combination and be able to detect it.
And adding with the snipers, that kind of security at the single base pair level, it's like limitless, even more.
Yeah. And so I guess to add to that also, so we're not at a point yet where the algorithm can perfectly predict the best performer. And so you probably saw in some of the slides where we've been testing, well, you saw, I told you or mentioned that we're building 24 sensors for each of these regions. And that is because we still need to do some empirical testing to make sure to find that top performer.
Okay, great. We have another question here from Greg that says, Dear Keith, thank you very much for the nice presentation. Do you think there would be some benefits in using your recently developed platform of electrochemical detection for COVID-19 diagnostics? So, of course, you did cover this slightly, in the second half of your presentation, but maybe just kind of re-emphasizing the difference between your technology and maybe some of those other tests that are out there.
Sure. Yeah. So Greg, thanks for reading our work. So we put out a paper in November where we used, instead of color-based reporting, we used an electrochemical-based reporter, this was work done in collaboration with Shayna Kelly's lab in pharmacy as well. And the motivation behind that work was so that we could detect multiple things in one reaction. And so to get at one of the earlier questions where you might want to stack multiple sensors in one reaction, absolutely. If we could multiplex sensing for COVID, like let's say a couple snipers and a couple more generic sensors in one test, that would be phenomenal. However, that technology is still very much early days, right? And so that's one of we're really motivated to build out into that multiplexing sort of direction. But we've purposefully picked our most tried and true sort of robust sensors for this project because we want to be able to make an impact and contribute to the crisis. And the best way to do that is to sort of implement what's already been established. And so that's sort of the rationale behind our approach.
Okay, our next question, I may mispronounce something here, but this one's coming from Rod. It says, are you using heat to lyse virus and provide access to sensors or something else?
Well, maybe I'll mention what we're doing now, and then Margot, maybe you could mention what you did in Brazil.
Sure.
So right now we're using synthetic RNA, which doesn't require any sample prep. When we go to our patient trial, we will be using RNA extraction kits, just like conventional diagnostics like PCR are using today. And that is because we want to validate, again, with the motivation of being able to move as quickly as we can and make an impact. We're trying to take the most like solid path to deploying these tests. But with reagent limitations and RNA extraction being a challenge, we are thinking about alternates. And so Margot's done some work in Brazil with Zika, and maybe you could share your work with there and how
Sure.
we solved it. Yeah.
So just to add on what Keith mentioned, like Health Canada doesn't require RNA extraction specific protocols for getting the genome of the virus out of the patient sample. So we could use quite a few things. In Brazil, I've tried to detect Zika virus into boiled samples. So instead of using a kit, instead of using resin or any type of chemicals, you just take your samples and then you boil it 5 minutes at 95 degrees. And then we did compare our Toehold sensor and the qPCR, and actually it was working better with the Toehold. We could get a factor of we obtained 10 times better sensitivity boiling than using an RNA extraction kit. So we also hoping that we'll be able to test that approach with Masood later on. Yeah.
Thank you. So we're coming close to the end of time, so we have time for one more question. I also just wanted to let everyone know that we will be able to, we're working on posting a recording of the webinar on our website, pharmacy.utoronto.ca, and we should also be able to share the Q&A here on our website as well. So just to let you know, if you wanted to share with colleagues, you can check back to our website in a few days from now. So I'm just going to jump to a question here. This will be our last question from Peter, who says, great talk, Keith, and nice rapid work, everyone. Are you still using Pure in the COVID-2 sensor? And if so, do you foresee any difficulties getting enough to mass produce diagnostics? So Pure is P-U-R-E. I don't know if that's how you pronounce it.
Yeah, that's absolutely. Yeah. So
great.
Actually, that's excellent. Good question. Absolutely. You know, so one of the advantages currently is that Pure Express, which is our reagent that we use the Toehold switch in, and it comes from New England Biolabs, is not in the PCR supply chain. And so it is not under the same strains and pressures as PCR reagents. And we've been in close contact with New England Biolabs to sort of basically ensure that we'll have enough reagent. And that's our plan, right? So yes, we are using Pure Express, and we are planning to continue to do that. But you can imagine there will be a break point where if we can scale our tests to what we think is actually needed domestically and internationally, we'll have to use something else. And so we have other cell lysate-based systems that can be manufactured locally, very simple equipment that should work as well, and that we've used, or I've used in the past. We're focused on Pure Express, but we certainly could expand that way down the road. Do you guys have any other final thoughts about Pure Express or cell lysates? Or
I guess we're trying to make an impact today, I mean, tomorrow. And then, yeah, development of the tools and that are needed to scale and to be even, even have cheaper solutions, are in development.
Yeah, to sort of add to that, that reminded me, so our sensor costs sort of all in right now is around $10 for a triplicate test. And this compares to a triplicate test, and these are rough, these are of about $50 for PCR. And so so we're already quite a bit less expensive than PCR. And for applications in low and middle income countries, switching over to this lysate-based system would reduce the cost by an order of magnitude, if not greater. And so that would be another motivation for switching over to these other systems at some point.
So we're just about 2 minutes after 2 now. So I wanted to offer an opportunity for any final thoughts from you, Keith, here, and also to thank everybody for joining us and for participating in the chat. We do have lots of questions here that we will post answers to on our website later this week in a few days. So Keith, I'll give you the opportunity to just close with any final thoughts.
Okay, sure. Thank you, Kate. Well, first of all, thank you and the team for organizing the seminar so we could share our work. We're very grateful for the opportunity. And of course, like I've mentioned, but I want to emphasize, thank you, Masood and Margot. You guys are doing amazing work. And I think the spirit that is motivating Margot and Masood is the same spirit that's sort of motivating the scientific community to respond to this outbreak. I've seen incredible creativity and collaborative work in the last, more than normal, right? It's absolutely incredible how collaborative the community has been in sharing reagents and sharing access to tools and sharing tools. And so our tools, these Toehold switches will all be shared publicly as soon as they're validated, so that others can use them. And so I think it's a great time sort of for science to shine in response to this challenge, all the way from sensors and PP and technology, all the way to vaccines and fundamental biology, understanding the infection process of the virus and so forth. And so I think we're going to see a huge leap forward in the coming months as these projects are published. And so it's a hard time for everybody, but it's also there's a lot of optimism and hope when I see the amount of sort of community spirit that's out there. So with that, I will sign off, but thank you very much. And yeah, Masood and Margot, do you guys have any last thoughts?
Just want to thank everyone for joining and listening to us, and looking forward to testing our platform with real patient samples and being able to distribute this as soon as possible.
Thank you, everybody.
Okay.
Thank
well,
you. Thanks, everyone.
Okay, thank you.
Everyone.